All right, just wanted to get into this 2013 paper I had on mixed and branch linkage polyubiquitin. We focused mainly on K48 and K63 linkages, but what's really cool is we came up with our own nomenclature system, which you can see on the bottom right of the slide. It's a great system with K48 and K63 because there's a lot of activity receptors and different properties that these linkages have that we can exploit to study how they function in the context of mixed and branch linkage polyubiquitin chains. The paper is pretty NMR heavy, but we have some interesting conclusions about how these affect going beyond homogeneous ubiquitin linkages. So with that, let's go dive into the notebook LM AI summary of the paper.
So today we're conducting a technical audit of the 2013 Nakasone et al. Paper. We're going to do a really results-oriented analysis, focusing on their investigation into the structure, the signaling, and, most importantly, I think, the synthesis of these mixed-linkage polyubiquitin chains. You know, this image really captures the whole problem they're tackling. What we've known for a long time, these homogeneous ubiquitin chains, well, that's just the tip of the iceberg. The real frontier and what this work is all about is this huge, uncharted territory of mixed and branched ubiquitin polymers. And that's where the potential for some serious signaling complexity lies. Okay, so here's our game plan for this audit. First, we're going to quantify the combinatorial challenge. Then, we'll critique their enzymatic assembly method. From there, we'll dive into the structural analysis using NMR and the data on receptor binding. We'll also look at the nomenclature system they proposed before we wrap up with a technical summary and what it all means for the field. All right, let's get right into our first point, just trying to get our heads around the sheer combinatorial complexity of this entire polyubiquitin system. This table really puts the scale of the problem into perspective, and this is pulled right from their supplemental table S1. So for just a simple tri-ubiquitin chain, you've only got 8 possible homogeneous linkage chains. But look at this. You have 56 unbranched and 28 branched mixed linkage possibilities. That's 84 total mixed linkage chains. They outnumber the simple ones by more than 10 to 1. This really drives home why the synthetic and analytical tools they developed were so badly needed.
So with that kind of complexity, how on earth do you synthesize these chains with any degree of precision? Let's really critique the experimental design and their synthetic methodology.
The whole secret to their control really boils down to 2 key tricks. First, they use lysine to arginine mutations on the distal ubiquitin. This K to R swap basically puts a hard stop on chain growth at a very specific lysine. And second, they used a D77 C-terminal deletion on the proximal unit. Think of it as a reversible block that they can just pop off using the enzyme YUH1. This 2-part strategy gave them absolute control over pretty much everything: length, topology, and the exact linkage.
Now this schematic from supplemental figure S1 shows exactly how they did it for unbranched chains. For a specific chain like this UB63 UB48UB, it's a step-by-step process. First, they make a dimer with one E2 enzyme, then they take off that D77 block with UH1, and finally, they bring in a different E2 enzyme to add the third ubiquitin. It's a really precise multi-step assembly line.
But here's where it gets really elegant. In stark contrast, making the branched trimer was incredibly efficient. They showed that if you just throw in 2 different linkage-specific E2 enzymes at the same time, one for K48 and one for K63, you can build the whole branched trimer in a single reaction. I mean, that is a huge methodological leap for generating these complex structures in the lab.
So the synthesis is elegant for sure, but how did they prove the final structure was what they thought it was? Let's dive into the nuclear magnetic resonance data they used to confirm the conformation. Okay, this data from supplemental figure S2 is absolutely crucial. They're looking at chemical shift perturbations, or CSPs, to compare the ubiquitin units in their trimer to standard dimers. On the left, you see hardly any changes for the K63 linked unit. That tells you its environment is pretty much identical to a normal K63 dimer. But on the right, the K48 linked unit shows big shifts, which is the classic signature of its compact shape. The takeaway here is massive. Each linkage in this mixed chain keeps its own native, distinct conformation.
And this right here, from supplemental figure S3, this is the smoking gun. This NMR spectrum is looking directly at the isopeptide bond signals on that central ubiquitin. You see the unique signal for a K48 linkage in red and the one for a K63 linkage in blue.
But in their branched trimer, shown in green, you see both signals at the same time. That's direct, unambiguous proof that they successfully created both linkages on a single ubiquitin.
All right, so the structure is confirmed. But what does that mean for function? Let's turn to the binding assays. We need to see if these chains can actually send out these mixed messages to the cell.
So check out this model, which they built from their NMR titration data. This shows a known K48 linkage sensor binding to the chain. And look, it latches right onto the proximal and K48 linked units, just like it's supposed to. But what's really critical is that the K63 linked Ubiquitin, over here in blue, is totally ignored. The receptor selectively reads only the K48 part of the signal.
Now, here's the flip side. You take that exact same branch trimer, but this time you introduce it to RAP80, which is a K63 specific receptor. The complete opposite happens. RAP80 grabs onto the proximal and K63 linked units, and now the K48 linked unit is pushed aside, basically invisible to this receptor. This proves that two totally different signals encoded in one molecule can be read independently.
And this quote from the paper really says it all. The structural and the signaling identity of each linkage is preserved. They don't get blurred together. A K48 linkage acts like a K48 linkage, a K63 acts like a K63, and that allows the cell to read both messages from a single molecule. You know, a really significant and maybe underappreciated part of this work is their proposal for a standardized nomenclature. I mean, given the insane complexity we saw earlier, the field desperately needed a clear systematic language to even talk about these things. They came up with 3 simple but really powerful rules. 1, directionality is always distal to proximal, left to right. 2, the linkage type is just a superscript before the receiving ubiquitin. And 3, for branches, you just pop the multiple distal units into brackets. This system finally gives us a clear, unambiguous way to describe pretty much any ubiquitin polymer you can think of.
All right, let's bring it all home with our final section, a technical summary of the findings and what this means for the ubiquitin field moving forward.
So, to summarize the key technical wins here, they developed a solid method for making these defined mixed linkage chains. They used NMR to prove that each linkage keeps its native shape, and crucially, they showed that different receptors can independently read their own signals on a single branch chain. They even showed these chains can be processed by the proteasome, which is an important functional check. And of course, they gave us that much-needed nomenclature. The paper's main conclusion is really a paradigm shift. It gets us away from this old idea of one chain, one signal. We now have to think of these things as multi-signal polymers. A single ubiquitin chain can act like a molecular switchboard, potentially plugging into multiple, even conflicting, downstream pathways all at once.
So in the end, this work gives us the foundational tools and the concepts to start asking completely new questions. It opens the door to this huge functional signaling library that was totally out of reach before. And it leaves us with this massive question. What new signaling outcomes, what cellular processes are actually controlled by this combinatorial logic? Answering that is going to be a major focus for the ubiquitin field for years to come.